After studying this module, you shall be able to know various parameters for determining water quality standards, learn the importance of dissolved oxygen, identify the purity of water sample by various tests, evaluate the biochemical and chemical oxygen demand. An adequate qualitative and quantitative water supply is most essential for small as well as big cities and towns. Along with water supply, then there should also be a means of disposal of wastewater. Disposal of wastewater is of widespread national concern. Municipal waste and industrial activities generate a large number and variety of waste products which are generally discharged into water streams. The situation is particularly critical in developing countries where there are no stringent laws to check that the industries treat the water generated prior to disposal. The analysis of the most common industrial solvent water is extremely important as it contains a large number of impurities which are necessarily to be checked before the water is used for any specific purpose. The ways in which water is used are so varied that no single purity standard can be applied. If water is to be used for cleaning or washing purposes, no special treatment is required. If water is to be used for drinking purposes, then it is most essential to determine color, turbidity, dissolved solids, hardness, alkalinity, acidity, free chlorine, etc. In order to control water pollution, quantitative measurement of extent of water pollution is extremely necessary. A specific treatment cannot be undertaken without knowing the substances and their quantities in water. The measurement of water quality is therefore very tedious process and a large number of quantitative analytical techniques are used for these purposes. We shall now discuss some analytical procedure for water pollutant analysis. In this module, we shall study three important parameters, that is dissolved oxygen, biological oxygen demand, chemical oxygen demand as an index for water pollution. First, we will study the dissolved oxygen. DO is one of the most important indicators of the quality of water for aquatic life. Dissolved oxygen analysis measures the amount of gaseous oxygen dissolved in an aqueous solution. Oxygen is necessary for all forms of life and it enters water by diffusion from the surrounding air, by aeration as rapid movement and as a waste product of photosynthesis. It is measured in its dissolved form as dissolved oxygen. Adequate dissolved oxygen is necessary for good water quality. Natural stream purification process require adequate oxygen levels in order to provide for aerobic life forms. When the dissolved oxygen levels in water drop below 5 mg per liter, aquatic life is put under stress. The lower the concentration, the greater is the stress. Oxygen levels that remain below 1 to 2 ppm for a few hours can result in large fish kills. Wastewater from sewage treatment plants often contains organic materials that are decomposed by microorganisms which use oxygen in the process and decrease the amount of dissolved oxygen of the water body. If more oxygen is consumed than is produced, dissolved oxygen levels decline and some sensitive animals may move away, weaken or die. DO level fluctuates seasonally and over a 24-hour period. They vary with water temperature and altitude. Cold water holds more oxygen than warm water, and water holds less oxygen at higher altitudes. Thermal discharges such as water used to cool machinery and in manufacturing plant or a power plant raise the temperature of water and lower its oxygen content. Dissolved oxygen levels in natural as well as wastewater depend on physical, chemical and biological activities of the water body. The analysis of DO is very important in water pollution control as well as wastewater control. Now, we will study about the determination of dissolved oxygen. Three methods which are widely used for the determination of DO are Winkler method, which is also called iodometric method or titarimetric method, polarographic method and the electrometric method, which use a membrane electrode. First, you will study the titarimetric method. It depends on the oxidizing property of dissolved oxygen. There are 4 different titarimetric methods based on water sample which is to be tested. Number 1, Winkler method. Number 2, azide modification. Number 3, alum fluctuation modification. And number 4, permanganate modification. However, in all of these methods, the basic principle remains the same. Then the choice of the method depends on the sample which is to be tested. Number 1, azide modification. In this method, the interference caused by nitrite is removed effectively because presence of nitrate causes interference in biologically treated effluent. Number 2, alum fluctuation modification. In this, if the sample contains relatively large amount of suspended solid, then this method should be used. Number 3, permanganate modification. If the sample contains ferrous iron, then this method is suitable. We will add 1 cm³ of azide solution that can be adopted to suppress the interference due to ferric ions. Now, we will study the titarimetric principle in detail. The chemical determination of oxygen concentration in water sample is based on the method which is first proposed by Winkler in 1888 and further modified by the Strickland and Parson in 1968. Oxygen in the water sample oxidizes iodide ion to iodine quantitatively. The amount of iodine generated is then determined by titration with the standard thiosulfate solution. The endpoint is determined by using starch as a visual indicator. The amount of oxygen can then be calculated from the titer value as 1 mole of oxygen react with 4 moles of thiosulfate. At the time of sampling, dissolved oxygen is first fixed by adding Mn2 ions under basic condition, which results in a brown precipitates of manganic hydroxides. Prior to analysis, the sample is acidified to a pH of 1 to 2.5. This causes the precipitate hydroxides to dissolve and liberating the Mn+3 ions. Now, Mn3 ions oxidize previously added iodide ions to iodine. The iodine is then titrated with thiosulfate. Iodine is reduced to iodide and the thiosulfate is oxidized to tetrathionate. All these equations are shown here. Mn+2 ion under basic condition converts into the brown precipitate, and then this brown precipitate of manganese hydroxide reacts with the iodide ion and iodine is liberated. The liberated iodine is titrated with standard thiosulfate solution, and the dissolved oxygen is measured by volume of the thiosulfate multiplying it with the normality and with the equivalent weight of oxygen divided by the volume of the water sample taken. Nitrite interference and azide modification. Nitrite in water sample causes interference by oxidizing iodide, as nitrite plus iodide they will give N2O2, which is then oxidized by oxygen entering during the titration. It results in enormous high value and the endpoint cannot be detected. So, it may be estimated by addition of sodium azide to alkaline solution. On acidification, NO2 is decomposed, as you can see in the equation. Now, this figure here explains you the process for the determination of dissolved oxygen. We will take a BOD bottle with the water sample. To this 1 ml of manganese sulfate is added, followed by addition of 1 ml of alkali iodide azide. Then this bottle is stoppered and it is shaken well. After that we will add 1 ml of sulfuric acid, again shake it and then transfer this solution to a titration flask. Using starch indicator, this sample is titrated using thiosulfate. Next method for determination of DO is polarographic method. This method is based on the fact that oxygen is readily reduced at the dropping mercury electrode. An aqueous solution saturated with air exhibits two polarographic waves which are attributed to oxygen. The first wave is obtained as a result of reduction of oxygen to peroxide, as is shown in equation, and second as a result of further reduction of peroxide into water. Thus, oxygen is polarigraphically active and is reduced first to peroxide and then to water. In water at 0.05 volt and minus 0.94 volt. The polarograms given by oxygen serve as a means of determining DO in water. Thus, in polarographic analysis, usually nitrogen is passed through the solution for several minutes just before the determination. In addition to dropping mercury electrode, oxygen can be reduced at several other electrodes in aqueous solution if a small negative voltage is applied. The magnitude of the current flowing through this solution can be determined by the rate of diffusion of oxygen to the electrode. This method is not used in the DO analysis of domestic or industrial wastewater because the mercury electrode get poisoned by impurities of the test solution. The third method which is used for determination of DO is membrane electrode method. In this method, two metal electrodes of silver and lead are dipped in a saturated solution of KHCO3 potassium bicarbonate, which is separated from the water sample by using a polyethylene membrane which is of 0.06 mm thick. Thus, a galvanic cell can be plugged to a pH meter to give a direct reading of DO in milligram per liter. The 0 to 14 pH scale in pH meter now becomes 0 to 14 milligram per liter dissolved oxygen. The current is measured for number 1 for the sample, number 2 for the standard which the sample after saturation with air, and number 3 for the blank sample after treatment with sodium carbonate to expel oxygen. Now, what happens at positive and negative electrode? At negative electrode, which is silver electrode, water get oxidized and lead will now react with the alkaline media to form the lead oxide, and at the electrode the lead in alkaline solution converts into lead hydroxides. Next parameter is biochemical oxygen demand. Biochemical oxygen demand or BOD is a standard measure of the amount of oxygen that would be needed by the microorganism to cause biodegradation of a part of organic and inorganic pollution load. The organic matter like starch and sugars are consumed by microorganism as food. The inorganic constituents like sulfides and ferrous ions are attacked by oxygen in the presence of enzymes released by microorganisms. Ultimately, the pollution load is transformed into simple non-hazardous compounds and dissolved oxygen is used in this process. The higher the BOD, the more oxygen will be demanded from the waste to break down the organics. The BOD test is most commonly used to measure waste loading at treatment plants and in evaluating the efficiency of wastewater treatment. The BOD test is performed by incubating a sealed wastewater sample for the standard 5-day period, then determining the change in dissolved oxygen content. The bottle size, incubation temperature and incubation period are all specified. Most wastewater contain more oxygen demanding materials than the amount of DO available in the air saturated water. Therefore, it is necessary to dilute the sample before incubation to bring the oxygen demand and supply into appropriate balance, because bacterial growth requires nutrients such as nitrogen, phosphorus and trace metals. These are added to the diluted water, which is buffered to ensure that the pH of the incubated sample remains in a range suitable for bacterial growth. Complete stabilization of a sample may require a period of incubation too long for practical purposes. Therefore, 5-day period has been accepted as the standard incubation period. The result is called the 5-day BOD and is expressed in milligram oxygen per liter of water or in ppm. Pure drinking water on the monthly average should have 5-day BOD in the range of 0.75 to 1.5 milligram per liter. BOD value of a raw sewage runs from 200 to 400 ppm. BOD values of several hundred ppm indicate strong sewage. The drinking water should have BOD less than 1 ppm. The environmental impacts of BOD are: number 1, sewage containing high BOD create environmental and health problems. Number 2, it interferes with the aquatic life. Organic pollution is harmful to fish as it tends to reduce the amount of dissolved oxygen. And number 3, it defines the strength of domestic waste and industrial wastewater. Now, you will learn the method for the determination of BOD. The BOD test relies on a measurable depletion of DO over a specified period of time. The BOD test is used to measure waste load to treatment plant, determine plant efficiency, which is in terms of BOD removal, and control plant processes. Because the test is performed over a 5-day period, it is often called as 5-day BOD or a BOD 5 written in subscript. In its simplest form, a BOD 5 test would involve putting a sample of waste into a stoppered bottle, measuring the concentration of DO in the sample at the beginning of the study and again 5 days later. The difference of DO value would be the BOD5. Most relatively unpolluted streams have a BOD that ranges from 1 to 8 mg per liter. If the BOD5 value of a sample is less than 7 mg per liter, sample dilution is not needed. A BOD5 value greater than 7 mg per liter requires sample dilution. Dilution is necessary when the amount of DO consumed by microorganism is greater than the amount of DO available in the air saturated BOD5 sample. BOD5 values are acceptable only if DO concentration after 5 days must be at least 1 mg per liter and at least 2 mg per liter lower in concentration than the initial DO. If during the 5 days DO drops to 0, the test is invalid. Therefore, it is always necessary to dilute the sample to keep the final DO above 0. A 5-day BOD of the diluted sample is given by DOI means initial DO minus DOF means final DO divided by the dilution factor. And how you calculate dilution factor? P, that is volume of wastewater taken divided by volume of wastewater plus dilution water. Now here you see an example for the calculation of 5-day BOD test for an unseeded sample, like a 10 ml sample of sewage mixed with enough water to fill a 300 ml bottle which has an initial DO of 9 mg per liter to help assure an accurate test. It is desirable to have at least a 2 mg per liter drop in DO during a 5-day run, and the final DO should be at least 2 mg per liter. For what range BOD5 would this dilution produce the desired result? Now you can solve this problem. You will first calculate the dilution factor P, which is 10 divided by 300. Now you know you have at least 2 mg per liter drop in DO. So BOD5 should be greater or equal to DOI minus DOF, which is equal to 2 divided by dilution factor, which gives you 60 mg per liter. And to assure at least 2 mg per liter of DO remaining after 5 days requires BOD5 should be less than DOI minus DOF, which is 9 minus 2 divided by dilution factor, and it comes out to be 210 mg per liter. So this dilution will be satisfactory for BOD5 values between 60 and 210 mg per liter. Now you will see the example of seeded 5-day BOD test. So far it is assumed that the dilution water added to the waste sample has no BOD of its own, which would be the case if pure water were added. In some cases, it is necessary to seed the dilution water with microorganisms to assure that there is an adequate bacterial population to carry out the biodegradation. In such cases, to find the BOD of the waste itself, it is necessary to subtract the oxygen demand caused by the seed from the demand in the mixed sample of water and dilution water. In this case, 2 BOD bottles must be prepared, one containing just the seeded dilution water and the other containing the mixture of both wastewater and seeded dilution water. The change in DO in the bottle containing just seeded dilution water, which is called the blank, as well as the change in the DO in the mixture is then noted, and using this equation the BOD of the waste water is then calculated. Now I will give you this solved example for BOD determination. A test bottle containing just seeded dilution water has its DO level dropped by 1 mg per liter in a 5-day test. A 300 ml BOD bottle filled with 15 ml of wastewater and the rest seeded dilution water experiences a drop of 7.2 mg per liter in the same time. Determine the 5-day BOD of the waste. So, first you will calculate the dilution factor, which is 15 upon 300, which will be 0.05, and using the formula given above, you will calculate the BOD of the wastewater, which comes out to be 125 mg per liter. The next parameter is chemical oxygen demand. BOD does not give accurate results of pollution load because non-biodegradable wastes like pesticides and detergents are resistant to microbial degradation and hence do not affect BOD. Some other pollutant like cellulose get slowly oxidized. Thus, during the 5 days period required for calculation of BOD, these may be only partially oxidized. Sometimes the water sample may contain toxic elements which may poison the microbes even before they can act on waste. In order to overcome these problems, another analytical parameter called COD or chemical oxygen demand is used. COD analysis is the measurement of the oxygen depletion capacity of a water sample contaminated with organic waste matter. Specifically, it measures the equivalent amount of oxygen required to chemically oxidize organic compounds in water. COD is used as a general indicator of water quality and is an integral part of all water quality management program. Additionally, COD is often used to estimate BOD as a strong correlation exists between COD and BOD. However, COD is a much faster and more accurate test. This involves a 2-hour digestion at a high heat under acidic conditions in which potassium dichromate act as an oxidant for any organic material present in water sample. Silver sulfate is present as a catalyst and mercuric sulfate acts to complex out any interfering chloride. Following the digestion, the extent of oxidation is measured through indirect measurement of oxygen demand via electrons consumed in reduction of chromium 6 to chromium 3. This can be done by titration or by spectrophotometrically. The water sample is heated with the known quantity of potassium dichromate for several hours and the unreacted dichromate is then determined by titration with standard Morse salt solution. Now, determination of COD in the reflux flask, add the wastewater sample, standard potassium dichromate and sulfuric acid reagent through the sides of the flask. Cap and mix the content and reflux for about 1 to 2 hours. Repeat the procedure by substituting distilled water for sample. Transfer the content of the reflux flask in the titration flask. Add distilled water to make the volume to 50 cm3. Now add ferrone indicators 1 to 2 drops and titrate against standard ferrous ammonium sulfate solution. From this equation, we will calculate the COD of the water sample, which is volume of the more salt used in the blank minus volume of more salt used for the sample multiplied by the molarity and divided by the volume of the sample taken. Next, total organic carbon. This is the best method for the measurement of total organic matter in the water samples. TOC test uses heat, ultraviolet light and a strong chemical oxidant or a combination of these three to oxidize the organic compounds to CO2 and water. Oxygen demand is measured indirectly by determining the amount of CO2 produced using IR spectroscopy, conductivity or calorimetry. This test can take several minutes to several hours to complete. Now, this table summarizes difference in BOD, COD and TOC. First, oxidant. COT use potassium dichromate or manganese sulfate. In BOD, oxidation is done by microorganism, while in TOC it is done by oxygen, K2S2O8, heat or by combination of the above. Now, most suitable use. COD is rapid and frequent monitoring of treatment plant efficiency and water quality. BOD, modeling treatment plant process and the effects of organic compounds on the dissolved oxygen content of receiving water, while TOC measures amount of total organic carbon in sample. Test completion time. COD requires 1 to 3 hours, BOD requires 5 days and TOC several minutes to hours. Advantage. COD correlates with BOD on waste with constant composition. Toxic materials do not affect the oxidant and it has short analysis time. While BOD, it most closely models the natural environment when used with proper seed, and TOC correlates with BOD on waste with constant composition but not as closely as COD and it has short analysis time. Disadvantages. COD has interference from chloride ions. Some organic compounds are not oxidized completely, while BOD has interference with toxic materials which kill microbes, and microbes do not oxidize all materials present in the waste and it is a time consuming process, while TOC require expensive equipment. Some oxygen compounds are not oxidized completely and it measures total organic carbon and not oxygen demand. Now, I will summarize what you have studied in this module. Alkalinity, acidity, color, turbidity, dissolved solids, hardness, free chlorine, BOD, COD, DO, coliform count, etc. are some of the parameters which are used to check the quality of water. Dissolved oxygen analysis measures the amount of gaseous oxygen dissolved in an aqueous solution. When the dissolved oxygen levels in water drop below 5 mg per liter, aquatic life is put under stress. The lower the concentration, the greater is the stress. BOD is a standard measure of the amount of oxygen that would be needed by the microorganism to cause biodegradation of a part of organic and inorganic pollution load. Pure drinking water on the monthly average should have a 5-day BOD in the range of 0.75 to 1.5 mg per liter. BOD value of a raw sewage runs from 200 to 400 ppm. BOD values of several hundred ppm indicate strong sewage. COD analysis is a measurement of oxygen depletion capacity of a water sample contaminated with organic waste matter. Specifically, it measures the equivalent amount of oxygen required to chemically oxidize organic compounds in water.